Okay, thanks, JB. I hope everyone can hear me in the room. Thanks, Jens, for the presentation before. That was really very interesting. I'm very sorry I can't be there with you today. Unfortunately, I had an argument with a metal motor vehicle as a pedestrian a week or so ago, and so I came off worse in this altercation. And I'm hence back in my kind of spare room, reliving the glory days of the COVID pandemic, speaking into a computer. So hopefully this won't be a good replacement for an in-person lecture, but hopefully it won't be too bad experience for all of us. Yes, as JB said, I'm going to talk about ocean biogeochemistry and primary production. But like Jens, I want to start a little bit by telling you my kind of background, how I got to this position. So I'm trained as a biologist. I did an undergraduate degree in marine biology in the lovely city of Newcastle upon Tyne in the northeast of the UK. I graduated in 1999, a long time ago, it seems now to me. And then I went on, I spent quite a lot of time actually trying to find the right PhD project when I started my career. During my degree, I got very interested in the Southern Ocean and the role of iron in regulating productivity in the system. And, really what I wanted to do was learn how to use these ocean models that Jens was mentioning in his presentation to try and test hypotheses and understand the importance of different mechanisms. And I had a lot of trouble trying to find a PhD where I could do that, largely because in the UK, as is common in most European countries, PhD is quite short. It's 3 years, maybe 4 years maximum. And that's quite a short period to learn new skills and then also use those new skills to go on and, make some new scientific discoveries that you need for your PhD thesis. So I went to do my PhD in the US, actually. I took a kind of random walk to Stanford University in California to work with someone called Kevin Arrigo, that some of you might know. And I worked on ocean biogeochemistry of the Ross Sea. And, this was a longer PhD. It took about 5 and a bit years for me to graduate, but it meant that I was able to, really pick up these new skills and take the time to expand my portfolio of interests as well. And so I graduated there in 2006. And then at that point, I moved to Paris to work with Laurent Bopp at the LSE, same place where Jens was. And I spent around 4.5 years in Paris, really working on global models, trying to understand the link between ocean carbon cycle, ocean productivity, and changing nutrient dynamics. And this was a really productive, fertile time. It was really exciting and enjoyable. Following that, I went down to a different hemisphere, so moving a bit further on the map, and I spent just over 2 years in Cape Town in South Africa, working with Pedro Monteiro, Seb Swart, who I guess isn't there anymore. He was there last week, I think, with you. Seb and I used to share an office many years ago, and this was a great opportunity to kind of work on modeling, but also work more on developing infrastructure, observation infrastructure, to measure iron and these kind of things in South Africa. And then I moved to Liverpool, where I took a permanent position in 2012, and I've been there since, and I was made full professor in 2018. And my kind of USP is that I use models. That's my kind of main skill set, but I'm a biologist at heart. I'm not a mathematician or a physicist, so that's not really my deep expertise, and so I really tried to position myself really at the sort of link between modeling and observational science, and trying to work across those two domains to really operate, I like to think, on some interesting questions and some interesting problems. As part of that, I've gone to sea quite regularly over my career. The picture you can see on the top left, I'm on the left, looks like I'm in a kind of witness protection scheme. That's Baptiste was there with me. That's where he and I met many years ago on the Aurora Australis, running a section from Hobart down to Antarctica. JB, I noticed that there's a new IPY in 2032-3, so we should try and go on a cruise together again, just to sort of mark this kind of decadal scale interaction we have at sea.
That would be lovely,
Al. The picture on the right
Whenever.
is my most recent time I was in Antarctica. This was on the Tara, where I was chief scientist on this sailboat that goes around trying to collect large amount of genomic information. And, you know, doing this is like really fruitful. It's a really exciting part of my work, and it's really where a lot of the new ideas are generated, really, from going to sea, observing the ocean, interacting with colleagues in this environment. And I think it's a really important part of the career of being an oceanographer if you're interested and able to do so. I've also worked a lot in recent years with the IPCC, so I was one of the lead authors of the Special Report on Ocean and Cryosphere in a Changing Climate, this ESROC report that contributed to the AR6, and that was very interesting. And one of the most interesting aspects was that I was one of the authors of the Summary for Policymakers, so I had to go to this in-person kind of approval session with all the governments in Monaco. When was that? Just before the pandemic, right? 2019, that would have been. And the figure, you can see a picture there on the bottom left of us on the stage. That's myself. Some, you know, you can recognize your favorite oceanographers, Mike Meredith, Thomas Frelicker, Bob Halberg. You know, in a huddle, trying to figure out how we are going to respond to some criticism of some sentence we have written in the Summary for Policymakers from a particularly interested government or set of governments. And that was a really interesting part of my kind of career progression, because it really made me realize about what really do policymakers care about in terms of the science we do. How can we articulate the science we do in a way that makes them interested in what we're doing, and interested, importantly, of course, to take action on the findings that we have. Okay, so hopefully that gives you a little flavor of myself, my background, what I do. So today I'm going to talk about productivity and biogeochemistry, and I'm going to, you know, these lectures, I've designed them very much as kind of two parts. And so today we're going to try and set the scene. I think there's a lot of, you know, there are a lot of physical oceanographers there giving you lectures, I think. So we're going to change pace a little bit and start talking about the living organisms in the ocean and what they're doing. And one particular thing is the importance of this flux of carbon linked to the net primary production. We're going to talk about some of the key governing processes, and I'm going to end by telling you what we think, what our best estimates are for how productivity is going to change in the future with climate change, with a focus on the Southern Ocean. Okay, I would say interrupt me. Normally, I think that would be fantastic, but I think it's pretty clunky on Zoom, so maybe it's best if we just do the same as Jens did, and we can just have a Q&A at the end, and if needed, I can pull up any relevant slides. Okay, so let's get started. So this is one of these, like, you know, lovely diagrams that we see all the time about the role of biology in the ocean carbon cycle. Tons of arrows, lots of kind of artistic interpretation being used to the maximum. You know, we've got fish, we've got the land-ocean continuum, you know, some particles, bacteria, even like breaking down bacteria into different forms, so the archaea as well. And, you know, this is very complicated. There's lots of processes going on. And so at the risk of not patronizing you, I'm going to show a slide that I showed for my undergraduate, the final year undergrad lectures here in Liverpool, which is where I just sort of simplify this figure into a very simple view about what's going on with the ocean carbon cycle, where you can see I've really extended my artistic limits in terms of my attempts to represent some of the key processes. But really what we're thinking about is the fact that there's some, you know, physical chemical processes that set an equilibrium between CO2 in the atmosphere and CO2 in the ocean. The biology intervene here. You can use your own interpretation of what I'm trying to represent. But there's a whole lot of ecosystem processing, and then there's a sinking flux of carbon that regenerates in the ocean with similar physicochemical processes going on, some linked to biological activity like fragmentation of particles by zooplankton, trying to garden, you know, curate a kind of mesopelagic garden, and ultimately the physics brings this all back to the surface. But within this, there's a key flux, and it's represented by this kind of arrow here, and it's called net primary production, and that's the one we're going to focus on, because without this arrow here, we don't care about the whole rest of the diagram, right? We'd be in an abiotic world, and physics and chemistry would rule. We would be able to use, physical laws and chemical laws, and we wouldn't have to worry about all the mess of the biological processes. But we don't, so we have to, okay? And so that's what we're going to worry about today. Before we get started, again, at risk of not patronizing everybody, but it's good to call an apple, and so we know what we're talking about. So there's some definitions we need to use here. So the first one is gross primary production. Okay, so this is the photosynthetic reaction, for those who don't recognize it. It's the combination of CO2, water to produce a sugar and oxygen, okay? So hopefully everyone knows that, or at least has come across it at some point in their career to date. What we're going to talk about today is what's called net primary production. And so that's this rate of photosynthesis, and we're going to subtract off the respiration. So some of these sugars are broken down by the photosynthesizer to fuel their cellular metabolism in the mitochondria. And so the balance between the two we call net primary production. In some places you might hear about net community production, which is this net primary production minus all the respiration by the entire phytoplankton community, by the entire ecosystem community. That might quite closely approximate export production, if you know about that. But we're going to focus here on NPP, okay? This is a critical process whereby, we might argue it's the most important biological reaction in the ocean, maybe the most important reaction. I don't know, if I were there in person, we could have a discussion about that over a beer or have an arm wrestle. But, using the energy in the sun, inorganic molecules are converted into organic molecules, and that's a really critical process. So what does it look like? We have a system we have an assay to measure primary production in the ocean, but it's not used in a very widespread sense, and we tend to rely on satellites to give us a measurement. So we exploit the fact that this absorption of light during the photosynthetic process alters the optical properties of light. Satellites can pick this up, and then some mathematics can be done, and that alteration to the optical signature of the surface of the ocean can be used to infer an abundance of chlorophyll, for instance, that you might have heard of, or the amount of productivity that is occurring. And so we see, high productivity in upwelling zones, high productivity along the coasts. If we look at the Southern Ocean, it's beautiful to see down at the bottom here, relatively low productivity offshore, higher productivity near the coast again. So again, I think, you really need to worry about this process. It's a really important process. It catalyzes almost all of the responses, all of the contribution the ocean makes to ecosystem services. If you're worried about ecosystem diversity, biodiversity, if you're worried about feeding people with protein from the sea, if you're worried about water quality and whether you have a harmful algal bloom or not, this process is at the nub of the matter. I totally agree with Jens. The contribution of the biological pump to the ocean carbon cycle is not massive. And I certainly agree with his assertion at the start of his lecture. But nevertheless, all of the biological contribution, however large it may be, is catalyzed by net primary production, as I showed at the start. If there was no net primary production, there'd be no contribution. And then, finally, it's an input for upper trophic levels. So if we want to forecast somehow the response of some member of the more charismatic community of the ocean, then we need to know this. This is the energy and organic carbon input to the base of the food chain. So again, at risk of not patronizing you, I'll go very quickly, but if you've never heard of this, photosynthesis is split into two reactions. They both occur within the chloroplast. This is a very simplified representation of that. The first are a set of reactions known as the light reactions because they need light. Okay, those occur in the thylakoid. There's a stack of thylakoids here. The important thing is that these produce two things. They produce ATP, adenosine triphosphate, energy, and NADPH, which is what we call reductant. So this is the energetic and chemical input into to drive a set of chemical reactions. These occur in a process known as the Calvin cycle. And we sometimes call these the dark reactions. It doesn't mean that they happen in the dark. It just means that they do not need light. Okay, so phytoplankton are still doing this in the light. It's just that they don't need light for this. And the Calvin cycle effectively uses the fruits of the light reactions, adenosine triphosphate, NADPH, to fix CO2 into sugars. Okay, the water splitting reaction that produces oxygen is in the light reactions. Okay, and if you want to see what it really looks like, this is an electron microscopic view of a chloroplast. And if you see these little kind of stacks here that look a little bit like kind of sort of burgers or pizzas lined up, they're the thylakoids. That's where this is all happening. That's where all this action is going on. So if you want to understand net primary production, we need to think about, what is it, first of all? So it's a rate. So it's some quantity of carbon, milligram per square meter here. It could be moles. You could choose per liter, per cubic meter, whatever. There's some amount of carbon per day, okay? And it's generally the product of some rate of growth or some specific rate of carbon fixation. In this case, this would be in units per day, and the biomass, standing stock. How many phytoplankton do you have? If we have one phytoplankton fixing carbon really fast versus 1 million phytoplankton fixing carbon very slowly, it's highly likely the million will actually fix more carbon in total. So we need to think about the standing stock of the community, okay? The first part is the growth rate, okay? And this is the kind of simple diagram of what a growth rate looks like. If you grow a phytoplankton in the cell, in the lab, or if you monitor their growth kind of at sea in some hypothetical situation, you would see this exponential growth curve of the cellular abundance or cellular carbon changing as a function of time, okay? And this rate of change in the concentration of the phytoplankton is driven by their standing stock and their growth rate, okay? And we can write this exponential out as an exponential equation. We can use the natural logarithm to linearize the equation, and then from that, the slope of the line is the growth rate, okay? So that's how we experimentally go about measuring the rate of growth. If you swap abundance here for amount of phytoplankton carbon, you'd be then approximating the specific rate of net primary production, okay? Because there'd be some autotrophic respiration. Many, many years ago, someone called Dick Epley, who passed away recently, collected a lot of data on growth rate. So that's what you're seeing here, growth rate in this kind of old-fashioned unit of doublings per day, but it doesn't really matter. Bigger number, bigger growth. And he plotted these as a function of temperature. And what we can see there's this upper limit set by temperature, this exponential relationship, which we call now a kind of Epley curve, which is linked to the role of temperature in driving cellular metabolism, worked on by Arrhenius many, many years ago. And then what we see is the data. So it's interesting that the data, none of the data passes the line, but most of the data actually falls below the line. So most phytoplankton don't actually achieve these theoretical temperature-constrained growth. And that's because of nutrient limitation, okay? That's because their ability to divide, their ability to build new cells is limited by the raw materials they need to build those cells, okay? If we want to move house, we need, and build a complete duplicate of our house, we need all the stuff that goes in the house, okay? And the phytoplankton are in the same boat. So they require nutrients for kind of two reasons. One is because they're structural, right? They make up the fabric of their cell. So a good example here might be the photosynthetic pigments that are made of nitrogen, or the phospholipid bilayer that forms the cell wall of a plant cell is made of phosphorus. Otherwise, they need nutrients to catalyze cellular reactions. Okay, so RNA, so translation of DNA, RNA to undergo replication is full of phosphorus, right? So you need a lot of phosphorus for this process. And the photosynthetic reaction that we spoke about just now, there's iron throughout that, so you need a lot of iron. And then it's the absence of these key nutrients will either limit your growth because you can't build new cells, or because you can't produce your metabolism effectively doesn't operate any longer. Okay, and if we want to look at nutrient limitation, again, this is a classic result from Dick Dugdale back in the late 60s, where he looked at the amount of phosphorus you would add to a cell culture and the oxygen evolution, right, as a measure of photosynthesis. And you see this kind of nonlinear response that looks very, very much like Michaelis-Menten enzyme kinetics. And you can describe this mathematically by some kind of maximum rate of enzyme activity, or in this case, photosynthetic activity, some half saturation concentration where you're at half of your maximum rate, and then you can very clearly link the rate of a process to substrate availability. So those models that Jens was talking about in his presentation, these are the kind of equations they're using to drive their model architecture. These, it's not just an equation, it represents real processes, right? When cells want to take up nutrients, so if we're a cell over here and we want to get nutrients into our cellular, intracellular space to put them to work. We've got two processes, that cellular transport occurs in the cell boundary layer. So we're limited on the one hand by the molecular diffusion of properties, solutes, into the phycosphere, the cell boundary layer, and then the ability of our transporters to internalize the nutrient in question from being in the phycosphere to actually being in the cell. And that's what explains this kind of non-linear curve, that you have an initial part which is diffusion limited, which is relatively linear, and then at some point your diffusion is high enough that you're diffusing enough nutrients to your cell boundary layer that your nutrient uptake is then limited by the amount of transporters that you have. So these mathematical abstractions are trying to represent real processes. So what nutrients should we worry about? Okay, and to do this, we can use a concept known as nutrient deficiency. So this is a very influential plot produced by Mark Moore in 2013, so just about 10 years ago. And what Mark did here is that he compared two quantities. He first normalized all the availability of all nutrients in seawater to the availability of carbon. Okay, so for a biologist, we put carbon as our kind of core currency. So he looked at how much nutrient there is, how much there is relative to the amount of carbon, and then he did the same for the phytoplankton cellular contents. Okay, so we looked at basically how much of a given nutrient is in the cell per unit carbon. Okay, and obviously carbon sits on this one-to-one position, and we use that as a reference point to look at the availability of other nutrients in seawater. Okay, so these are all bioessential. Phytoplankton need these. The black line represents the one-to-one ratio. So anything up in this quadrant, you would say there's way more of it in the ocean than the phytoplankton will ever need. Okay, so even if they grew for an infinite amount of time, they would never deplete the amount of those nutrients. Okay, good examples here are the salts in seawater. So magnesium, for instance, if you like chlorophyll, the center of chlorophyll is made of magnesium. But there's way more magnesium in the ocean than phytoplankton will ever need. Magnesium will never become limiting. Same for sulfur, potassium, strontium, molybdenum. And it's only when we get into this area below the line that we start to get nutrients that are less abundant in seawater than they are in the phytoplankton cells. Okay, and those are called deficient. Okay, so a deficient nutrient isn't limiting. It has the potential to become limiting. Okay, if there's enough phytoplankton growth. And so we can see nitrogen and phosphorus, kind of as expected. Below the line, silicon also for diatoms. And then we have a whole load of transition metals here down in the bottom corner that are at very low abundance in seawater, orders of magnitude more scarce in the ocean, and orders of magnitude less essential in the cell. But nevertheless, there's not enough of them in the seawater to support infinite levels of phytoplankton growth. So they have iron, manganese, cobalt, that's cadmium, copper, and zinc. And nickel is kind of hovering. Okay, so this bar represents the major uncertainty, is the fact that for many of these elements, the phytoplankton requirements can be quite plastic. Okay, so if we want to worry about nutrient limitation, it's in this quadrant that we want to be. We're not going to spend much time worrying about the strontium availability. And the eagle-eyed amongst you, hopefully you have this on a big screen and not JB's laptop, is the red colors here represents elements that have residence time on orders of thousands or tens of thousands of years. Okay, so they're very abundant in the ocean. The ocean stockpiles them, they make up the salts of the sea. And it's those elements that have shorter residence times that eventually emerge as becoming limiting in the ocean. Okay, so what does nutrient limitation look like in the ocean? This is a paper that came out last year by Tom Browning, Guillemard, and Mark Moore in Southampton. And what they've done here is they've compiled all the experimental determinations of nutrient limitation that exist in the ocean. Okay, so everywhere where you see a circle on this map, somebody has stopped a ship, they've collected seawater from the ocean, and they've conducted what's known as a bioassay experiment. And there's a sort of graph, a sort of example kind of experiment here, and I'll show you some more easy to interpret ones on the next slide. But effectively what the scientists do is they add different nutrients alone and in combination with one another, and then we measure some component of the system, here chlorophyll, right, taken as a proxy for the phytoplankton biomass. And we see which nutrient did we have to add to get the biggest response in terms of chlorophyll. And then you color the circle depending on what that nutrient was. And those colors are all here. And in general, we see this high-low latitude distinction. Okay, so the low latitudes, let's say the Atlantic for an example, most of the low latitude Atlantic is blue, okay, and so you had to add nitrogen to drive a response in the community. And the Southern Ocean, which is what we're all interested in, that's why we're here at this summer school, you can see it's virtually exclusively red, okay, and that means iron. Only when iron was added did you see any response in the phytoplankton community. The backdrop represents two things, okay? So the colored shadings with the greens here, that represents the surface nitrate from World Ocean Atlas. So we see, obviously, the high nutrient conditions of the Southern Ocean because of the upwelling, as Jens mentioned. Then you have North Atlantic, North Pacific, Equatorial Pacific, also relatively high nitrogen systems. And then the blues represent where the nitrate concentration is very low. And then the shading, this kind of reddish color, is an interpretation from a model of the aerosol deposition of iron, so the input of iron from the atmosphere. And so you see it's basically when you don't have iron, or you do have loss of nitrogen, or vice versa, that's when these nutrient limitation patterns emerge. Okay, you may have so this shows a kind of like complex interpretation of the circle with some serial and co-limiting behavior, and you can see some of this behavior in the Southern Ocean. There's a split circle there, there's a kind of halo here around this one in purple. In the Benguela we see a lot of this behavior. In the West Pacific and near South China Sea we also see a lot of this kind of split behavior, and I want to just mention that briefly for you and also show you a little bit of what these experimental results look like. Okay, so this isn't the Southern Ocean, apologies for that. This is the Benguela upwelling system. It's a paper we worked on with Tom a few years ago. And I just want to show you the results of 3 of these experiments so that when you may never use these again, but at least when you hear about them, you'll understand where they come from, okay? And so experiment 3, let's find it on the map. It's here, right, near the coast of, I think we're up near Angola there. And this shows you the result of this experiment. So Tom conducted a multifactorial experiment adding iron, cobalt, nitrogen, and vitamin B12. And C is the control. And what we see is in this experiment, it was only when nitrogen was added that we saw a statistically significant increase in the amount of chlorophyll. And then when we added nitrogen and cobalt, nitrogen and iron, and nitrogen, iron and cobalt, nitrogen, iron and vitamin B12, we could never statistically increase the chlorophyll above this level. Okay, so in this scenario, it would be singly limited by nitrogen. It's only nitrogen that we need to worry about. The circle is blue. And then we have what's called singular and serial limitation. Okay, so this is experiment 6. This is just off the upwelling here. And in this example, we see again, nothing really happens until you add nitrogen and cobalt, nothing happens. But then when you add nitrogen and iron, you get a massive increase in productivity. So we call this serial limitation. This is when, yes, there is something singly limiting the system, but when you add enough of that, something else quickly becomes secondarily limiting or serially limiting, okay, so in series. And then the final is what we would call co-limitation, okay, so in this situation, we had cobalt, nothing happens, iron, nothing happens statistically, iron and cobalt, nothing happens, nitrogen, nothing happens either. So this is unusual. When we've added either iron or nitrogen, nothing's actually stimulated this community. It was only when we added iron and nitrogen in combination with one another that we saw an increase in productivity here. So we call that co-limitation. Okay, so apparently this community needs both nitrogen and iron in order to grow. And that makes life a little bit complicated to understand why that is. Okay, so this co-limitation, a large area of the ocean is co-limited. The Southern Ocean, I don't doubt, is similar in this regard, and we'll see a little bit of evidence for that tomorrow. And there's 3 kind of types of co-limitation that can occur. The first is what we call independent co-limitation, and that's purely because the concentration of 2 resources are being simultaneously drawn down to low levels in the ocean. Okay, so an example of that might be where nitrogen and phosphorus get drawn down together to very, very low levels. You have to add both of them back together to get above their limitation thresholds. The other types of co-limitation are a little bit more kind of interesting, maybe a little bit more complicated. The first is this substitution co-limitation. And this links back to these catalysts that I was talking about. Many of these catalysts, they use metals as cofactors, but many of them can actually use different types of metals in these enzymes. So they may substitute one for another, which leads to co-limitation. So for instance, you may be growing in the Southern Ocean, in the Ross Sea, the mixed layer may be very, very shallow. You may have drawn down carbon to very, very low levels. And now you need this enzyme, carbonic anhydride, to convert carbon. And you're worried then about the fact that this enzyme can use either cobalt or zinc. Okay, so you may be co-limited by these elements because this enzyme can operate with one cofactor or another. Another example is that within the kind of light reactions of photosynthesis, we'll see in a second, many of them require iron, but many of them can be substituted for other metals. Okay, and so you can use, instead of cytochrome C6, you can use plastocyanin, which uses copper. So we can generate this dependency between elements purely around the fact that different enzymes can operate with different elements. The other is what we might call a dependency, okay? And so there's a very subtle difference, I think, between these two, but it's worth thinking about it. So the dependency is when your ability to acquire one nutrient is driven by the availability of another, right? So for instance, if you are highly stressed for phosphorus, the most common form of phosphorus in the ocean is phosphate. If you've depleted all of that in organic form, you may want to acquire what's called dissolved organic phosphorus. So this is phosphorus in organic forms. But to do that, you need an enzyme. Okay, you need an enzyme to basically snip the phosphate atom off this complex organic molecule. That enzyme is called alkaline phosphatase, but that needs metals. Okay, that needs cobalt or zinc. So it's possible that your scarcity, if you don't have enough cobalt or zinc, you can't build alkaline phosphatase, and then you are more phosphorus limited. Okay. Another possibility is these transporters that we spoke about. One of the main transporters that phytoplankton use to get iron in the cell uses copper. So if you don't have enough copper, then you can't produce this transporter, and hence you're more vulnerable to iron limitation. Okay, and so we have lots of these interactions amongst elements at the individual level that can be quite complicated to disentangle. And I think in particular, if you want to understand the response of the system to change, we really need to worry about these different acclimatory strategies and what they mean for the community. The last point, I guess, is to make the point that when we measure nutrient limitation, so for instance, these slides, it's all chlorophyll here. Apologies, I've kept the label off the axis here, but this is chlorophyll in milligrams per cubic meter. And so it's possible actually that no individual organism is actually co-limited. But it's just that there's a community of different organisms, that one is limited by iron and one is limited by nitrogen, so that you get this community level co-limitation, okay, that responds. And that may mean that you say, okay, we don't need to worry about co-limitation because actually each individual organism is only limited by a single nutrient. But on the other hand, if we're interested in the impacts of the community and we want to resolve the impacts of the community, then we may need to worry about this, okay? So with this context of nutrient limitation, I want to go back to the Southern Ocean, okay? And so the Southern Ocean, as you see, is all red. If you zoom in very carefully, you can see a little bit of like half shading here that we'll talk about tomorrow. There's a little bit of vitamin B12 here in the Ross Sea that may be important, but by and large, it's limited by iron. These crosses represent the places in the ocean where the iron enrichment experiments have been performed. So that was the Soirée, Sofex, some of the German experiments, Lofex, Isonex-1, Isonex-2, where iron's been added. Hence they're a cross because it's a slightly different type of manipulation experiment. So if you want to worry about NPP in the Southern Ocean, we want to worry about iron, basically. I think you'd say it's the most important thing. So when I was an undergraduate PhD student, that was what we were all worried about. And the reason we need iron is because it is essential to the light reactions of photosynthesis. So this is more of a biologist's view of the photosynthetic reactions. It's photosystem 2. This absorbs the energy of a photon gets into an excited state. This energy cascades through a bunch of like redox catalysts, hits the photosystem 1, which is the second photosystem in the chain. It then gets boosted up to a higher energy level, and we cascade through another set of redox catalysts. Okay, that's how this cascading through the catalyst is how the ATP and the NAD pH is produced. And what you see is that every single one of these has iron in it. So this, the QA, the cytochromes are all full of iron. This is where the plasticine copper iron switch around cytochrome C6 can come in. These are all full of iron. The photosystems themselves are full of iron. Okay, so chlorophyll doesn't have any iron in it, but don't let anyone tell you that chlorophyll has iron in it. Magnesium is the cofactor of chlorophyll, but the reaction center that pigment is coupled to does have a lot of iron. So photosystem 1 has 12 iron atoms in every photosystem. Photosystem 2 has 1 iron atom. So you've got order of 20 odd atoms of iron involved in the photosynthetic process. And this represents roughly 70 to 80% of the cellular demands for iron. They also need iron for respiration, for the respiration process, and for the acquisition of nitrate. Okay, so the Southern Ocean is a very high nitrate system. So if you're acquiring nitrate as a cell, you can't put that to work in your cell until you've reduced it to ammonia. Okay, and to do that, use an enzyme called nitrate reductase. That needs iron. Okay, so we think that the changing iron supply in the Southern Ocean is going to be critical in terms of driving the growth rate changes that we might have seen in the past, that we might see now, or that we might expect to see in the future. Okay, and we'll dig a little bit more process-wise into this tomorrow. I also want to talk about loss processes. I don't know how much these have come up in the lecture so far, but we tend to focus a lot on the kind of what we call the bottom-up factors that modulate the growth rate. But as I mentioned at the start, if you can increase or decrease the total amount of phytoplankton in the ocean, what we call the standing stock, you're also going to affect the amount of NPP that you see. You're also going to affect the type of particles that may go on and fuel the biological pump. And many of these loss processes also are very important in terms of the fact that they can recycle nutrients. Okay, they can bring nutrients from the organic particulate phase back into the inorganic phase. And we know for iron, for instance, this is very important. The first one I want to talk about are viruses. Okay, so much like human viruses can make us stay at home and not travel to places and interact over Zoom, there's a big problem for phytoplankton as well. And they weren't really known about or remarked upon, until really this seminal paper by Jed Furman in the late 90s. And this is what you're looking at is an epifluorescence photomicrograph. And you can see I've marked a kind of protist here, pennate diatoms, and prokaryotic cells, and all these little small dots are viruses. Okay, so the ocean is full of viruses, right? And there's tons and tons of them. And when we measure their, genomic sequencing from expeditions like Tara, for instance, we can find lots and lots of viruses in the system. And to give you a clue as to how important they may be in the system, this is an experiment that was done in the North Atlantic. Apologies not having a Southern Ocean example. And this is where they've tried to estimate the viral contribution to mortality and compare it to grazing, right, herbivory, which we might have instinctively thought would be the main term. And you can do this in the ocean by doing sequential, what are called dilution experiments, where you dilute the system with seawater of different cutoff sizes, and so you then basically introduce to your phytoplankton the presence or absence of viruses, and you just see how the mortality of your experiment changes. And in the blue bar, you've got the estimated mortality rate due to grazing, and in the red bar, you've got the mortality rate due to viruses. And this is just to show that the viruses are as important as grazers in terms of reducing phytoplankton biomass. What's interesting, I guess, or important to think about is that they may be very different to grazers in terms of what dying, if you're a phytoplankton and you die because you've been infected by a virus, you've lysed, is a process called viral cell lysis, where the cell breaks apart and effectively all the internal parts of the cell are released directly into the seawater. It's obviously very different if you get consumed by a secondary consumer, and then of course their metabolism comes into play. And that's why we're also interested in grazing as a removal agent of phytoplankton, and a modulator of the standing stock. And there are kind of, a bunch of different processes that we need to think about in terms of grazing. First is this ingestion process. How does the grazer ingest their prey? Do they use filter feeding? Do they use raptorial feeding? There's this concept known as what's called sloppy feeding, which is kind of, I'm sure you all consume your lunch in a very tidy manner. But some zooplankton, when they consume, they don't actually ingest all of the prey that they try to consume, and some of that gets released. And so you may ingest this amount of POC, organic carbon, but the amount of carbon that gets into the biomass of the grazer is going to be roughly reduced by about 10-15% because of this eating mechanism. Of that carbon that gets into the zooplankton, parts of that will be allocated towards the growth and reproduction, and parts of it will be consumed by respiration of the organism and by what's called excretion. And this excretion process is interesting because nutrients are released along this process pathway as well. And obviously, if a zooplankton is performing a lot of respiration, a lot of excretion, it's going to have what's called a very low gross growth efficiency. Right? So the growth efficiency is effectively how much of what you eat actually ends up in your biomass to produce new zooplankton. And then finally, we get this process known as egestion, which is the fecal material. You might have heard of fecal pellets. These are packaged, very dense, fast-sinking parts of the egestion from the zooplankton. And so these are really important in terms of understanding, what gets taken from the seawater into the phytoplankton, then is processed by the ecosystem. Okay, either zooplankton or bacteria. Some of that ends up in their biomass, some of it gets packaged up and exported into very fast-sinking particles, and some of it gets remineralized back to fuel more productivity in the system, okay? Let me check how we're doing for time. Okay, great. So if we want to understand what's going on with productivity, we need to think of a bunch of different factors, right? We've got these, what we call bottom-up processes, temperature, light, nutrients, that may affect the growth rate. Then we have from the top down, we have these grazers and other mortality feedbacks that may affect the standing stock. And then we've got the product of the two, the growth rate and how many phytoplankton you have, that sets the overall rate of net primary production, okay, that you might be interested in for a variety of reasons. I want to end by talking about what we know about the projections of net primary production, how we think this key process that links chemistry, biology, and some physics, how that actually might change in the future, and come now really down into the sort of Southern Ocean, the region of interest, to set the scene a little bit for tomorrow. Okay, so to make projections, we use models, as Jens mentioned in his presentation. We use these global scaled coupled climate models. This is a really nice web page. If you go here, Carbon Brief, how do carbon models work? It's a really nice article that talks you through the sort of historical evolution of the climate models and where they've been and where they're going. What this shows you is basically when different types of models were developed, in what decades, and at what stage they were integrated into the kind of Earth system models that we use to make forecasts today. So you can look at this and find your favorite thing. You can see ice sheets came in relatively late, as did marine ecosystems. Marine ecosystem models existed for many, many years, but it took decades for them to become integrated into the global climate models. On the other hand, the carbon cycle or the atmospheric chemistry came online much earlier. Okay, so before we look at the MPP, I want to just show you, what we see in terms of physical variables in the system. Okay, so this is sea surface temperature. These are projections of SST changes for the CMIP-6, so this is the Coupled Modeling Comparison Project, the 6th phase. These SSPs are the different climate scenarios, bigger number, bigger radiative forcing in 2100. These show you the CMIP-5 kind of analogous, but let's focus on CMIP-6 here. And this is effectively the surface average SST compared to a reference period. The solid line is the multi-model mean. The shading represents the spread of the models, okay? And so what we see firstly at the global scale, we see for SST, we see this really clear separation into scenarios, right? The main thing that dictate the SST at the end of the century is really the scenario that we end up on, okay? And then the multi-model spread is quite small, right? We can clearly pick out the green, the blue, the red. And this is really clear information for policymakers, right? Putting that IPCC hat on. And the more carbon we emit, the higher the climate change scenario, the higher the SST will be. This maps out at regional scales, right? And so here there's a measure of kind of model agreement. It's kind of stippling. So the stippling is where the models agree, and this stippling effectively, it's quite a low bar for model agreement. It's basically the majority of the models agree in the sign of the change, okay? So unsurprisingly, the ocean gets warmer, all models can agree that it gets warmer. It gets even warmer at the higher emissions scenario. Okay, and the reason I'm showing you this is that we can contrast this to what we see for net prime production now. Okay, so this is the same subset of those models, right? But still a fair few, tens of models. And now we see a very, very different picture, right? The global scale, we now can't pick out the scenarios as clearly as we could for temperature to contrast it with this, because the multi-model spread is now so large that it's swamping the scenario variability, okay? To the point at which the multi-model mean for many scenarios is near zero, plus or minus a very large standard deviation. Okay, the reason that exists is because none of the models really can agree on the directionality of change. So if we look here, this is the low emission scenario, SP126, or even at the high emission scenario, 585, we see very little stippling, okay? What that means is that most of the models don't agree on the sign of the change. Okay, so this presents a very different message to a policymaker. This says that basically I can't even tell you if productivity is going to go up or down in the future. Okay, so that's really stark level of uncertainty. We see similar things for other aspects of the biological system. We won't worry about that. And you might even argue this doesn't even represent probably the true uncertainty, because actually many of these models are actually very similar, and they all simplify many aspects of the marine system. If we look at regional scale, the story doesn't really get any better, right? So this is a regional analysis of different biogeochemical provinces. So now the global's down in the corner here. And what we can see is that there are, some regions where the uncertainty is enormous relative to the global uncertainty. So on the previous slide, you might have been struck by the global trend. So sorry, if we go back to this slide, you may have been struck by the fact that this envelope is enormous, okay? But if you look at different regions, that envelope can be 4 or 5 times higher, okay? So for instance, if you go to the Atlantic subpolar gyre, we may agree that productivity is kind of going down, but the 1 sigma uncertainty, which is a relatively conservative estimate of uncertainty, can't really tell us by how much things are going to go down. If we look at some key regions like the Equatorial Atlantic, the Indian Ocean, North Pacific subtropical gyre, South Atlantic subtropical gyre, again, we can't constrain the sign of the change, and the regional uncertainty is perhaps double or triple what we see at the global scale. So we've got a real problem here. The contrast is actually the Southern Ocean, okay, which is maybe what we're all worried about. And you can imagine we spend a lot of time trying to worry about what the Equatorial Pacific, why it's uncertain, or why the Indian Ocean is uncertain. I'm not sure that's much of interest maybe to this audience. So let's look at the Southern Ocean. And actually the Southern Ocean presents a really unique trend in the fact that the NPP is projected to increase, and the models seem to be relatively confident. If we go back to this slide again, you can see it's one of the only places where there's any stippling on the map, okay? So this perspective that the Southern Ocean is becoming more productive with climate change is very, emerges a very high consensus from climate models, okay? And that presents, again, an interesting message for policymakers. It suggests that we have a good understanding of what's happening in the Southern Ocean, right? That we project that the Southern Ocean is going to become more productive, more fertile. The analysis of this is basically because more iron is coming in from to the Southern Ocean from various means, and that's fertilizing the growth rate, as we saw, which is leading to higher levels of net primary production. Okay. So I'm going to stop. and we're going to pick this up tomorrow. But before we do so, I wanted to give you some kind of take-home messages. Firstly is that net primary production is maybe the critical carbon flux in the Earth system. If we want to understand what's going on with it, we've got to worry about some nitty-gritty of chemistry and biology. We need to understand how the changing nutrient requirements and nutrient availabilities and all these complex loss processes linked to things like viruses or even larger things like krill may come into play. Iron is unusually important in the Southern Ocean, so that's interesting, or you could say annoying, because it's an element that has very complex chemistry. It has a very short residence time, and so there's, it's not so easily controlled by the physics. And lastly, that the projections that we have now from our best available models suggest that the Southern Ocean is going to become more productive due to climate change, and that these models have relatively high confidence. This is the message we saw at CIMIC 5 as well. So the last kind of 10, 15 years, it's been quite a clear message coming out of the community. So tomorrow, we're going to dig into that. We're going to assess whether we are actually as confident as the models are in terms of these projections of NPP in the Southern Ocean. In doing so, we're going to look really into some more unique processes and unique aspects of the Southern Ocean and what makes it actually very different from a biological perspective to the rest of the world ocean system. And with that, I think I'll stop if we haven't gone on too long, and I'm happy to answer any questions that you might have.